So the second step is attaching firmly and negotiating the microbiome. So some of the ways, obviously, if the microbe is going to cause damage, most of the time it has to attach to the host tissues. So it can, once it's attached to the host tissues and survives the host defenses, it may produce some toxins and things like that, but in order to really cause damage, it has to be able to attach. So the second step, you have some sort of adhesion. So fimbriae, which are for attachment, they look like little cilia, but they're not for movement, they're for attachment. Capsules in bacteria, surface proteins and viral spikes could be, surface proteins could be on any kind of microbe, and then viral spikes, that's how they get into the cells. So they have some sort of adhesion molecule like that. So what is adhesion? Adhesion is a process by which microbes gain a more stable foothold on host tissues. And it's a lot of times you have to have the molecule on the host and a molecule receptor receptor on the host and a molecule on the microbe. And they have to be specific, but sometimes not. And a particular pathogen is limited only to those cells to which it can bind to. So if it can't bind to it for whatever reason, then it's not able to infect it. So once attached, the pathogen can invade body compartments. And so there's this idea of quorum sensing, which is that the bacteria produce chemicals. These chemicals communicate that they are at a certain critical level that's needed to establish infection. And they will not, they kind of fly under the radar until they reach that level, and then they start producing these toxins and things. Before that, they're not going to raise any eyebrows, they're not going to raise any suspicion with the host defenses, and so they will have this quorum sensing thing that once they get a high enough number of microbes and you have a critical level of microbes, then they can establish infection, and so they will then start producing toxins and things. So the adhesion molecules like the fimbriae here, capsules, that'd be another example, and then the spike proteins that adhere to a receptor on the membrane of the host tissue. And here are just some examples, and again, this table is only for informational service. This table is for only So here's a table of adhesive properties and different microbes that have those adhesive properties. It is not something, it's only for informational purposes. It's not something that's going to be on the test. So just FYI on that. Don't go through memorizing it because it's not going to, that you should know some examples, but you don't have to know which microbes have those examples. So the third step is then surviving host defenses. So once they have come in the right portal of entry, they have a high enough infectious dose, they have attached to the to the host cells, they have to then survive the host defenses, and that's specific and non-specific. So if there is no specific immunity, that's one way they can survive host defenses, but there's still probably going to be some non-specific immunity like phagocytosis. And so the big target here is avoiding phagocytes or avoiding phagocytosis in general, and then others will avoid the death inside the phagocyte. So they can somehow survive the chemicals that are in those phagolysosomes. So phagocytes are white blood cells that engulf and destroy pathogens by means of enzymes and antimicrobial chemicals. They basically eat them up, chew them up, and spit them out. And we've talked about this in chapter 14, so you should be somewhat familiar with the phagocytes at this point. Anti-phagocytic characteristics, they have virulence factors that are used by pathogens to avoid phagocytes. So there's something that they do, something that they produce that will help them to avoid the phagocyte. So it could be a toxin that kills the phagocyte. It could be they have a capsule that allows them to avoid phagocytosis, but somehow they circumvent some part of the phagocytosis process. So causing disease, this is step 4. So the 3 in green here, again, you're going to have to list these steps and explain what that means or give examples. Like in this one, it's just pretty obvious. It just means they adhere, and you need to be able to give some examples. But with surviving host defenses, be able to list a couple of different ways that they survive host defenses. So this 4th step is causing damage or disease, causing disease. And so we'll look at the enzymes or toxins, inducing excessive host response and causing epigenetic changes in the host chromosome. So the enzymes, you can see here, enzymes are secreted. The exoenzymes are used to break down their food externally, and the process of doing that allows them to invade further. So these enzymes, that's a big thing. Toxins are also produced, and they can do some damage, but those are, again, virulence factors. So if they produce those toxins or enzymes, then they're able to cause disease more easily, and without them, they do not. So they're structures, products, or capabilities that allow the pathogen to cause infection in a host, the adaptations that the microbe uses to invade and establish itself in a host, and they determine the degree of tissue damage that occurs. And so all of those are characteristics of a virulence factor. So direct damage using enzymes. Enzymes are exoenzymes. Bacteria, fungi, protozoan, and worms secrete these, and they're active outside of the microbe's cell. They break down and inflict damage on tissues. They break down host defenses, defense barriers, and that allows them to spread deeper into tissues. So some examples like mucinase is going to break down mucus. Keratinase is going to break down keratin. And hyaluronidase, which is this chemical hyaluronic acid, is a connective tissue. It's like present hyaluronic acid. So hyaluronidase breaks down this connective tissue, which is found, it's kind of like considered the glue between cells. And keratin is found in skin and nails, and again, helps to prevent microbes from getting in. So if you break it down, then the microbes have an easier access to get in. And mucus is one of our barriers to protection. So if you can break down the mucus, they can get in more easily. So toxins can actually be an enzyme as well, but toxin literally means poison. It is a specific chemical product of microbes, plants, and some animals that is poisonous to other organisms, and it helps get rid of the competition, as we have talked about before. But an exotoxin is one that is secreted by living bacteria cells to infected tissues, to the infected tissue. And there are many types. They're very specific to the tissues, and they're very like localized effects, where the endotoxin, not actively secreted and is shed from the outer membrane. Typically, it's a lipopolysaccharide, and it's only found in gram-negative bacteria. So exotoxins can be either gram-positive or gram-negative, and it's secreted, where endotoxins are part of the cell envelope, and only bacteria like gram-negative bacteria will produce them. So here's a table, and this is a table that I do want you to be paying attention to, and it is something that you'll want to know and be able to compare and contrast exotoxins to endotoxins. So a couple of things are, I'm going to highlight some of the things that are probably important, like effects on the body, toxicity, the chemical composition, fever, and then typical sources. Those are going to be probably the main ones that I'm going to ask about. So toxicity, if it's in small doses compared to high doses. So exotoxins are in small doses because usually these are enzymes, exoenzymes also. And so you only need one or a little bit because it reacts over and over and over again, where endotoxins are toxic in high doses, so you need a lot more. And the effects on the body is just really just general fever, and inflammation with the endotoxin, where the exotoxin is very specific to the blood or the type of cell. So like blood, liver, nerve. So there's like neurotoxins and hepatotoxins and blood toxins and things like that. And they will just be localized kind of effects. So the chemical composition, generally proteins, enzymes, so they're proteins, and lipopolysaccharides from the cell wall is what we see for endotoxin. So exo is a small protein, and endotoxin is usually lipopolysaccharides. And then the fever stimulation, usually not for the exotoxins. Yes, that's mainly what they do with endotoxins. And the typical source could be gram-positive or gram-negative for the exotoxins, but it's usually only gram-negative bacteria that will produce endotoxins. So make sure you can differentiate between exo and endotoxins. This will most likely be a matching question. So a couple of examples, like hemolysins are toxins. And so you remember the alpha, the beta, the gamma, hemolysins, or process of hemolysis that we looked at in lab. So inducing injurious host response. So this to me is a whole COVID thing, because it's the microbes themselves are not producing the damage. It is the host reacting excessively or in an inappropriate way in response to the organism. And so the pathogenicity is not solely determined by the microorganism. Pathogenicity is a consequence of this interplay between the microbe and the host. So the pathogen is causing injurious host response. And that is what COVID does. So, and a lot of viruses, this is how they cause damage. They don't produce toxins, they don't produce enzymes, but they do cause the immune system to overreact. And it's the actual symptoms are caused by the cytokines that your immune system is producing as it is fighting off the infection. So the inflammatory response and the cytokine storm that is produced in COVID is what causes the damage and what causes the disease in COVID. So that's a good example. So there are various types of diseases and a lot of terminology. So we're going to flip through these next few slides, and I'm just going to give you some terms. And so you need to kind of know and be aware of these. And these are in my alphabetical list of terminology that you need to know for this chapter. So an infection can be localized. It can cause damages locally and on a specific area of the body. It's confined to a specific tissue. So like boils, furuncles, carbuncles, those are all in specific areas. Fungal skin infections and warts, those are all localized examples. Systemic infections go all over the body, and you have several different systems involved. That's why it's called systemic. So it could be just one system, like it's all throughout the body, through the bloodstream, but it typically affects more than one system. So several sites and several tissue fluids, and it's usually in the bloodstream. So viral infections like measles and rubella, chickenpox, AIDS, bacterial infections like brucellosis, anthrax, typhoid fever, and syphilis. Fungal infections like histoplasmosis, coccidioidomycosis, or cryptococcosis. So these agents, they travel by means of the nerve system or cerebral spinal fluid. They can also travel through the nerves or the cerebral spinal fluid. So a focalized infection, a focal infection is one that starts in one location and is carried to other tissues. So a lot of times you get these, and this is the example they're giving, is this triangle of like the nose and the mouth area, and there's a lot of blood vessels here. And so this is why it's so critical that with people who have knee replacement or other surgery where they have a artificial joint or something They'll have to be on antibiotics for the rest of their life, or they're going to need to have them specifically when they go in and do dental work. So focal infections, moving from the mouth to the heart, because there's like this direct route, is a big thing. Tuberculosis, streptococcal pharyngitis like scarlet fever, toxemia. So the infection remains localized until the toxins are carried through the blood, and then they spread throughout or spread to a different target tissue. Mixed infections, you have several agents themselves that are simultaneously at the infection site. So a polymicrobial disease like gas gangrene, wound infections, dental caries, which is like plaque and like tooth decay, and human bite infections are all considered mixed. So you have various microbes. Primary infections are an initial infection, and then a secondary infection occurs after you've had the primary infection. So let's say your primary infection was the urinary tract, and that could then spread to a secondary infection in the vagina. So chronic versus acute. Acute comes on rapidly, has short-term effects. Chronic is going to progress and persist over a longer period of time. So it takes a little time for it to establish itself, and then it even persists over a longer period of time. So that's a chronic infection. You may have a chronic infection like hepatitis is one. Hepatitis B, for example, would be a chronic infection, and herpes simplex 2 or herpes simplex 1, those are chronic, long-lasting. I'd actually call the herpes, though, more of a recurrent infection, but anyway, chronic infections like hepatitis B would be one. Signs and symptoms. So these are warning signs of the disease. So you have damage. This is the damage section. And so signs and symptoms would be observable evidence, that would be a sign, and then subjective evidence that's only sensed by the patient. So you can't really measure a symptom, but you can measure or observe a sign. And a syndrome is a complex of signs and symptoms that identifies this as a particular disease or syndrome. So here's some signs and symptoms. So fever, we can measure it. Septicemia, we can detect it in the blood. Microbes in fluid tissues, we can detect it. Chest sounds, we can hear that. And skin eruptions, we can see that. So all these things are measured or observed. Symptoms like chills, well, that's just something you can't necessarily sense that. It's only sensed by the patient. They feel cold. Malaise, fatigue, chest tightness, nausea, vomiting. Well, vomiting, you can see that, but nausea, you can't. Abdominal cramping, lack of appetite, sore throat. And by the way, anorexia nervosa, where you don't want to eat, that's a psychological kind of thing. When we say anorexia in other terms in this respect, it is a lack of appetite. So a lot of times when you have an infection, you have a lack of appetite. You don't want to eat. So sore throat would be another one. It's something you can detect or you sense as a patient, but they can't measure or see it. So inflammation, has fever, fever, redness, swelling, loss of function, heat, all that. Those would be signs and symptoms of inflammation. And so you have the edema, which is the fluid, and you have granulomas or abscesses. And those are, granuloma is a walled-off collection of inflammatory cells, and the abscess may have bacteria in there as well with pus. So pus lesions with pus are abscesses, and granulomas pretty much without, but they're kind of walled off areas. Lymphangitis would be swollen lymph nodes. And signs of infections in the blood. So whenever you have increased white blood cells or decreased white blood cells, that's called leukocytosis if it's increased, and leukopenia if it's decreased white blood cells. Those can indicate infection. Septicemia, you have bacteria, lots of bacteria, and they're multiplying. Bacteremia is just present bacteria, small numbers of bacteria, but they're not necessarily in high numbers, and they're not multiplying. That would be the difference between bacteremia and septicemia. Just the presence of viremia, whether they're actively multiplying or not, is viremia. So infections that go unnoticed, we have different terms. So they all kind of mean the same thing: asymptomatic, subclinical, inapparent. Basically, you're not manifesting. You have no symptoms. And if the patient experiences no symptoms or disease and does not seek medical attention, so that's like subclinical, they're not seeking medical attention. And most infections are attended by some sort of sign.